Welcome to Office Hours Unplugged, where we chat with the most prominent researchers and PIs at McMaster. Today we have Dr. Ray Truant joining us. It's an absolute pleasure for you to come down here and join us. Would you be able to introduce yourself to the audience?
Sure. So I'm Ray Truant. I'm in the Department of Biochemistry at McMaster, and I'm a full professor in that department. And I've been at McMaster since 1999. My lab is almost entirely focused on Huntington's disease, but we're interested in neurodegeneration in a broader scope of diseases as well.
That's cool. Okay. Can you walk me through how you got into academia from the very beginning? I mean, how did it all start? What was your
So I did an undergraduate at University of Toronto.
Oh.
And downtown campus, I had no idea what I wanted to do, so I was doing a general life sciences-based degree. And in third year, I got very interested in virology and microbiology, because at that period of time, the HIV epidemic was hitting pretty hard across North America, and literally it was very analogous to what happened in 2020 here with COVID, right? So everybody wanted a solution, and students would just, outside of our classrooms, talk about what could be possible solutions for this virus. How does this virus work? Why is it different? Why is it so elusive? Why don't vaccines work? So I got very interested in that, and I finished a degree in microbiology. I was not an A student by any means, but it was good enough to get me into grad studies. And I continued at U of T in the Department of Medical Genetics, which was a very powerful department at that time. There were a lot of prominent researchers cloning genes. So this was also ingrained in my undergrad, is that what made U of T famous at that period of time is we had a lot of very successful gene hunting labs up and down University Avenue. Wow. So we cloned a lot of important disease genes. That made the press in a big way. It made the cover of journals like Nature and Science. These people were very famous.
Wow.
And I'm not sure why Toronto did this so much better than anyone else at that period of time. But unfortunately, U.S. Ivy League paid attention to all of this and poached everybody away.
Really?
So all of these researchers essentially ended up in the Boston area at Harvard.
Wow.
or Massachusetts General. But one of those researchers was David Hausman, who actually, in a very short period of time, along with Marcy McDonald and Jim Gazella, McDonald and Gazella are Canadians. They moved down there. They ended up cloning the gene for Huntington's disease in 1993.
Wow.
Very impressive.
But that was before I had any interest in that disease.
Yeah.
So I did my graduate work in the medical genetics department working for Jack Greenblatt, who was well known for his bacterial work at that period of time in understanding transcription regulation mechanisms. And he was using some very unique biochemical methods to determine new protein-protein interactions.
Yeah.
But when I came into the lab, he sold me on a project involving this new protein that he says, there's not a lot of people publishing on this, and it's called P53. And I was like, and it has a lot to do with cancer. And I was like, oh, that sounds fun. So I agreed to do the project. And of course, P53, which was actually originally identified in Toronto as well.
Really?
Yeah, it was actually the members of my committee. So my committee had the discoverer of P53 and the first person who characterized P53. And then this was my project. And of course, P53 papers went from about 22 a year to a little over 3,000 a year in about 6 years. And it was the central focus of cancer research for a long time. And it's still a focus of research, right? So, of course, in all of that, I didn't realize that it would roundabout to me years later. So I finished my doctorate, and but I was still interested in virology, and the HIV problem was still a big problem. There was not drugs that were working.
Yeah.
The biggest problem is that there were drugs, but people were getting resistant to the drugs very quickly. So there were 3 or 4 drugs that were out there that all hit different parts of the replication pathway, but the patients would become resistant to any one of the drugs.
Yeah.
So we needed more molecular mechanisms. So I went to go work with Brian Cullen at Duke University in North Carolina. That's at the Howard Hughes Medical Institute.
Uh-huh.
So I was a Howard Hughes Research Fellow for 3 years with him. And he really taught me how to run a lab, how to manage people. Everything about his lab was a group effort with all kinds of collaborations going on with other labs. So there was never a single author paper. It was always a group of authors working together.
Wow.
And he really understood how to manage projects to get the results. And what impressed me about him at that period of time is that there was a tremendous amount of research in HIV getting published, very similar to COVID.
Yeah.
So when I saw COVID coming in 2020, I immediately realized this was a complete parallel to what had happened with HIV, which means you're going to see a lot of research, a lot of research funding, and a lot of very bad research.
Right.
Yeah.
And Brian's work was always reproduced by others. Wow. And so he was very trusted as a researcher in HIV. But by the end of my postdoc period, triple therapy had come out of David Ho's lab in New York. So basically he did the math and he said, well, we give each one of these drugs to these people and they become resistant. But that's because the virus has a very fast replication rate and it also has a very high error rate. So he did all the math and he said, what happens if we give them 2 drugs? They're still going to become resistant. But if we give them 3 drugs, they shouldn't be resistant.
Right.
And that's exactly what happened. So triple therapy started in the late 90s, and it's been the therapy until today, more or less. The people have tuned the pharmacology of it.
Yeah.
And of course, lately, HIV is really getting controlled by the PrEP protocol, which is essentially completely protective against getting the virus.
Wow.
So, but in 99, Brian made the conclusion, we're done with HIV. Like, there's triple therapy out there. It's about vaccines now. It's not going to be about molecular mechanisms. But I learned a lot about basic molecular mechanisms of how proteins move in and out of the nucleus in the cell, and how HIV does that and how it's similar to other normal proteins and non-viral proteins. And I was like, this is going to be what I'm going to focus my lab on.
Oh.
And he was totally supportive of that. And I just said, I need to find a disease of interest.
Yeah.
And then it's a bunch of serendipity. So I'm at a pub with my friends who are all researchers at Duke. They came from all over the world. So for whatever reason, I gravitated to the British postdocs.
Mm-hmm.
Yeah.
And there's a Scottish postdoc at the pub one night, and he said he listened to like my story about I need a focus to start my own lab, and he said, you should look at Huntington's disease. There's a weird thing where the protein accumulates in the nucleus, and they have no idea how. And he gave me an example of a grant that he wrote to this agency in the U.S., and it got rejected. But he said, I think they would be really amenable to funding this kind of work. So this period of time, I was recruited by McMaster, but I actually didn't tell them what the exact focus of my lab was going to be, which is very unusual. You wouldn't do this today.
Yeah.
But I sat down, and before I showed up, I wrote grants to the U.S. agencies, and I wrote grants to what was the CIHR back then, which was the Medical Research Council of Canada.
Oh,
And everything got funded.
wow.
So I started hitting the ground running, and I came in, and one of the things that I had learned was the importance of live cell imaging and live cell biology
Yeah.
in my postdoctoral work. So I sitting there in my lab, and I brought in these guys from Nikon and Zeiss, and I said, I want a microscope to do live cell imaging with an incubator, automated capture, tracking, computer-controlled stage. I need all this kind of stuff because I had seen the scopes like this
At Duke?
at Duke.
Oh.
But what I didn't realize is these were not turnkey. These were all made.
Oh, wow.
So they basically told me, we can't help you. And I'm like, you're saying if I would need this, I have to make it myself? And they're like, basically, yeah. So I said, okay. So I learned how to build a scope up from parts, right? Trial and error, mistakes, lots of mistakes. It turns out to be very difficult to image live cells and keep them alive for a long period of time. But within 5 or 6 years, now there's turnkey systems you can buy, and it makes it a lot easier. But I got, so starting my funding in Huntington's disease, I said, well, this is what I'm going to focus on. And the main reason I focused on it was because I did have interest in Alzheimer's disease, because there was a very large group at Duke at this period of time that had, they were convinced that the cause of Alzheimer's disease was ApoE. And the stories I heard was, they've been moved from Duke over to Glaxo Laboratories in Research Triangle Park, and these guys have the cure for Alzheimer's disease. This is 1998 or something, right? So, long story short, that didn't work out at all. But I looked at the problem and talked to some Alzheimer's experts. I talked to some neurologists, and the first question is, what causes this disease? And the answer is, we don't know. We don't know for 99% of the patients. There's a genetic component for a very small number of them, but for everyone else, we have no clue. I bounce over to Parkinson. Same answer. We have no idea what causes this disease. So I'm a molecular biologist, biochemist by training. I need a protein to work on, right? So, and I looked at the bigger scope of it, and I said, we don't understand neurodegeneration at all.
Yeah.
And 30 years later, I would argue we've made incremental gains, but we still don't really understand it. Mm. But if you're going to really understand the disease, you need a disease that is monogenic. You need one disease protein to cause the problem. And if we can figure that out, we might learn a lot about neurodegeneration in general.
Right.
So you then look at the scope of monogenic neurodegenerative diseases, and Huntington's is one of them. It's not the most common, but it is the most funded for some reason. So you have to go where the funding is. That's the nature of science.
Oh, yeah.
And I've been supported by U.S. agencies since 2000. Mm-hmm. The Hereditary Disease Foundation out of New York, the Huntington Disease Society of America out of New York as well. For a long time, I was traveling back and forth across the border with these agencies. I also worked with the CHDI Foundation, which is based in New York, but is actually located in Princeton and Los Angeles. So I did a lot of traveling between LA, Princeton, New York, Hamilton.
That must have been a tough.
Well, it was the best way to describe this was an unusual situation.
Yeah.
In 2003, I'm called into the office at a meeting with a bunch of people I've never seen before. And they said to me, what would happen if we had a major donor who was extremely wealthy and willing to dedicate unlimited funding to this disease? And I said, well, that would be a nice fantasy, but that doesn't happen. And left the meeting, and then I found out it was happening.
Wow.
So it really felt like at that period of time the equivalent of a Manhattan Project, right? Because if you talk to any scientist in Canada, they'll always tell you, oh, if we only had more money, we could do this. And there's unfortunately not a huge priority in governments to fund research. I don't think that matches the priority of voters on both sides of the border, Canadians or Americans.
Yeah.
I think Canadians would love to see their tax dollars spent on research.
You think?
Because they all know they're going to get sick, or they all have friends who get sick, or at this period of time, everybody knows somebody who has had cancer or who has a neurodegenerative disease. It's, grandparents. It's not uncommon. So they understand that if you don't research this, nothing is going to happen, right? So... but this was a Manhattan Project. It basically said, we're going to centralize. They had a management structure. They worked with, at one point, over 600 different private contractual research organizations and academic labs.
Wow.
So they had their tentacles out all over pharma, all over biotech, all over academic labs, and they're doing this massive coordinated effort.
And this was what year?
So the around 2005-ish.
Oh.
Right? And so this is 20 years later.
Oh.
And so where are we now? In the early 2000s, we would go to meetings and they would be almost all academics. There was no industry interest. The reason why there was no industry interest is because they didn't see a viable target that they could work with. So the research symposium were all amongst basic scientists, more or less. But 2025, if you were to go to a Huntington's meeting, it's flipped around completely. It's almost all industry people. Mm-hmm. A lot of this was a change in how pharma sees the business of neurodegeneration. Originally, they were like, who cares about a disease that affects 1 in 12,000, right? You're not going to make enough money here.
Yeah.
If we'd have to charge a fortune for a drug. Well, then companies came along like Genzyme, and they made a therapy for Gaucher's disease, 1 in a million incidents, and they charged a lot of money for it. And it worked as a business plan. So now pharma is saying, well, yeah, we can make money on rare diseases. And then the Orphan Drug Act got passed in the United States, which protects the intellectual property around this research for much longer. So there's more money to be made. So now there's a business plan. If there's a business plan, pharma will come. And now we're at the position where we have about half a dozen different therapies in clinical trials, and a few of them are at the phase 3 stage.
Wow. So
we are imminently, in terms of a few years, going to know what is working and what isn't.
But in regards to Huntington's?
Yeah, in regards to Huntington's disease, right?
Wow.
And they're all working on one similar concept. We have this mutant protein that's getting expressed in people. By one way or another, we're going to try and lower the expression of this protein. And they have a whole new series of drugs called splice modulators that can do this. These are a new class of drugs. This has never existed before. But there are success stories in other diseases. So this is how the field is going. And then it comes down to how we're going to deliver these therapies. Is it going to be by a virus, or is it going to be by a pill? Is it going to be a chemical? That is going to be worked out.
Yeah.
So you look at this and go, well, I have a dream situation where someone tells me there's infinite resources to work with. Then now, 2025, people are saying, well, there's probably going to be a drug in the next 2 and 3 years, right? And I'm looking at this going, well, this was a dream literally 25 years ago. So be careful what you wish for.
Yeah.
So we now look at some of these other genetic disorders that do have a drug, and the drug is effective, and there are people worldwide affected by this disease, and the pharmaceutical company wants anywhere between $500,000 a year, or with a viral system, $2 million for the therapy per patient.
Oh my
Per
God.
patient?
Per patient. So this might work in the US if you have extremely good health insurance. In Canada, the Canadian formulary is covering some of these treatments because they are rare, right? So, but there's a question about how sustainable this is.
Yeah.
So remember I said Huntington's is a 1 in 12,000. That's what we thought 10, 20 years ago.
Yeah.
That number is likely 1 in 3,500 now.
Really?
So we're getting more and more information about the incidence of this disease. We're looking in different populations. We spent far too much time doing all of human genetics in European populations only, ignoring the rest of the world. And we're getting a better idea now of how widespread these diseases are. We had no information about Huntington's disease in India or China, South Asia, Africa, nothing, literally nothing.
It was just limited to like North America.
Yeah,
And then
and so people would actually quote, in reviews and say, well, Huntington's disease primarily affects Caucasians. And it's like, well, it doesn't. It actually affects everybody evenly across the planet. We just didn't pay attention to those populations, which is what human genetics is learning in terms of research, that this has been a long-standing problem. And that now there are CROs, these research organizations, that are looking at worldwide genetics, not just subpopulations in one region of the planet. And we're getting different information from those other groups of people.
Yeah.
So the challenge now is going to be, if we get a drug, how are we going to afford it?
Yeah.
And do we have to really rethink the model of how we make drugs? The model is we have academic labs that are doing basic research. At some point, they will land on a molecule or a regimen that may work, that spins out into a biotech company. I mean, I've done this. And then that biotech company will develop it further and at some point get bought out by a pharmaceutical company that will then have the money and expertise to carry out a clinical trial. But everybody wants their money back. But they want their money back with an anticipated return on investment of 500% in 5 years. So they want to make big money. Right? So this is a model that is in existence, but it is a flawed model. Because at the end of the day, do you really have a drug if that drug costs $2 million?
You might as well not have one.
Yeah. I mean, what is the point, right?
Yeah.
So on the other hand, if we know that something is working, if we know absolutely that lowering Huntington using small molecule splice modulators is the way to go, then we can think about government efforts to make a made in Canada version or open sourced version where we end up in something that is different than intellectual property that is out there, but something that could be a real drug because we could give it to people at cost or just slightly above cost to recoup the research costs with this back to the government. But in the end of the day, it could save the Canadian government billions of dollars.
Yeah.
Mm-hmm.
The current system of just like we wait for a drug to get approved and then we just pay out of the formulary, as more and more of these drugs come down the pipe, is just not sustainable. So then you got to start making decisions on which drug do you cover and which drug don't you cover.
Yeah.
And in some cases, they're going to be hard accounting decisions where you look at a person and say, if I put this person in long-term healthcare facility, or palliative care, it's going to be cheaper than giving them the drug. So
probably not better for the patient, but
it's not going to be better for the patient. So then, and then the other question that's coming up a lot in recent years is, just because a drug is approved by the FDA, does it really improve the life of these people on the drug? And the answer is not always yes. But if you have a family or an individual that is affected by this disease, I mean, their lives are horribly affected. They want some kind of solution. So even if it doesn't work, the families are going to be insisting that they have the ability to try it. And they will provide political pressure to get this to happen.
Mm-hmm.
Yeah.
And it really is a bit exploitive of this community, and people are willing to grasp at straws because they are in a desperate situation. So you completely understand why the community feels this way.
Yeah.
But on the other hand, if we start approving drugs because people want something, that's very dangerous.
It's a dangerous road to go down.
Because we're not making drugs that work. And of course, once the first drugs are approved and they start selling them, that completely shuts down research at the biotech level. They see the market has already been filled. They'll bounce to another disease. Lots of diseases to work on, right? So we need a different plan. We did a different model at this point of how we make drugs. And we need to think of a way that still involves business, that can still involve profit, and still involve recovery of costs, but in a way that's a little bit more manageable.
So you think that maybe people should think about that more closely next time when they consider political candidates and
yeah, unfortunately, this stuff never really becomes part of political campaigns. I have never heard a candidate for prime minister mention the word science.
Yeah.
In the entire time I've been back in Canada.
Wow.
Yeah.
It doesn't seem. I think it's one of these things where taxpayers just assume that's where their money goes. But I think they would be surprised at how much money we spend on healthcare in Canada and how much money we spend on research.
Wow.
And the answer is 0.4%.
Wow.
0.4%?
0.4% of what we
insane.
spend. We spend a lot of money on healthcare in Canada, right?
Yeah.
But we spent 0.4%
That is very
to discover
ludicrous.
new therapies, which could presumably save us a lot of money in that bigger pot of healthcare spending.
Yeah, and it would, in the long run, save the government, like you said, money.
Yeah.
It'll be much better for the people, I'd imagine.
But you need a government with a long-term vision beyond just 5 years and getting reelected again. And that's unfortunately not the model of politics.
Yeah.
But, we can hope, and we can eventually, every once in a while, we have a prime minister who does listen.
Yeah.
The John Chrétien government, they created the CIHR, they created the CFI Foundation. That changed the landscape of science in Canada.
Really?
So it could happen again.
Yeah. Wow. Oh well. I think we're about to wrap up here. Once again, thank you so much for coming on. It's been an absolute pleasure to have you on. You're very nice to listen to. You have a lot of great ideas, and thank you for coming on.
We'd love to have you again as well.
Yeah.
Sure.
Yeah, we have plenty of questions. So.
Yeah, unfortunately,
I wish we could do
I've been
this.
talking to you about the higher order stuff without talking to you about the nuts and bolts of what my lab actually does.
Oh yeah.
And what I've been doing at McMaster with our imaging facility here.
Oh yeah.
So yeah, definitely I'll come back.
Yeah,
This has been fun.
It's Office Hours Unplugged for a reason.
Yeah. You're probably the ideal guest to have on. Not much input is required for you to.
Oh yeah, I can go on forever.
Yeah. Yeah, it's just an infinite like store of information, experience. Well, yeah. All right. Thank you for coming on.
So much.
Thanks.
Yep.